Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the webinar section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speakers your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speakers, Samantha Hulla, PhD, Vice President, Biologics CMC, and Suneet Mehdi, PhD, Vice President, Biologics CMC, both from Syngene.
Thank you. So welcome to our presentation, De-risking Biologics Development, where we'll be talking to you today about practical strategies that you can employ to move faster from cell line to clinic. And I'll jump right into it. I'd first like to introduce Syngene real quickly as a contract research, development and manufacturing organization. We're headquartered in India. We support over 400 clients from all corners of the globe. We perform all services from drug discovery through CMC development and manufacturing in an integrated manner. With a workforce of over 8,500 people, we're very proud of our sustainability initiatives in particular. And I have included here that we were awarded Times Magazine Most Sustainable Company in India in 2025, which I think is really great. Next up, biologics. So we dive down. I mentioned that we were 30-year-old CDMO. We've been practicing 15 years of biologics services. We've been supporting our partners with CMC drug development and clinical and commercial manufacturing services for over 15 years. In that time, we've completed 150 projects successfully, enabled over 25 INDs and performed 250 GMP batches. My final slide on Syngene. We perform these biologic CMC services from 3 sites across India and the US. We have around 50,000 litres of drug substance manufacturing capabilities spread across bioreactors and fermenters from 200 litres to 4,000 litres. And we're well positioned to support our customers who are developing animal and human health products. The facilities that we have in Bangalore here on the right are additionally able to support drug product manufacturing as well as viral clearance studies. So hopefully I've given enough of an introduction to Syngene that allows you to understand the perspective that we bring to this topic of moving quickly from cell line to clinic. So let's jump right into it. As a CDMO, we speak daily to a large number and a wide variety of people and companies engaged in drug development. A common question that we hear over and over, the answer is sought by all, is how do we get to the clinic quicker? So taking a drug to the clinic meets that key inflection point in the valuation of that drug or that asset and the probability of it coming to market. It passes 3 critical milestones to get to that point. 1, is the drug manufacturable? 2, is it safe for animals? And 3, is it accepted as a new drug or an investigational new drug by the health authorities? At each of these milestones, data is collected by the developer, and each data package stepwise increases the value of the drug and also reduces the risk associated with its development. There are many hurdles on the road to reaching the clinic, however, including raising sufficient capital to fund the studies and gaining approval from a board to spend the money on this drug in particular, or to spend it on these studies in particular. The hurdles that I've mentioned don't discriminate in company size either. We find that biotech companies generally face both of those hurdles, raising funds and gaining approvals. But even large pharma can get stuck at seeking approvals to spend the cash that they have. It's worth remembering that reaching each of the milestones that I mentioned grants access to additional funding and likely to more approvals as well. So each of those milestones is critical. We prepared this presentation to share with you some strategies that Syngene employs that we've evolved to jump these hurdles consistently, but we've categorized them into 3 pillars that I'm showing on the screen now. Firstly, a business strategy. So how do we partner with our customers in a more helpful and efficient manner? A technical strategy. So how can we make the science work for us? Or to use Syngene's tagline, how can we put science to work? And then an operational strategies pillar. So how can we employ operational manners to cut time from that traditional timeline of 18 to 24 months to the clinic to a much shorter timeline to get there? So let me finish up my slides by talking about the MAB accelerator. So this is a business pillar, or one of the items in the business pillar that I mentioned. The MAB accelerator is a way of partnering with our early phase clients, with early phase drug developing customers. It satisfies the premise of moving quickly, but without compromise, and through these 3 key mechanisms. So 1, time to development start. Syngene commits resources really early on, and by this I mean scientists and equipment, and we'll commit those resources to begin cell line development immediately without financial commitment from the client or from the customer or from our partner. Cell line development allows that drug developer to achieve data around the question, is this drug manufacturable? Our customers therefore become or reach a much stronger position to gather funding and to gather those approvals. And Syngene makes this commitment without upfront payments, either through, for example, deferred payment terms or through success-based models, which leads me into that second line there, risk distribution. So risk is really high at the early stage of drug development, and free capital is usually very low within that small biotech. Syngene shares the risk with our partners by agreeing upfront what success might look like during cell line development. So for example, that could be a specific titer, goal, or a specific product quality. Our partners only pay if we hit that success criteria. So that's one of the ways that we can partner as part of this MAB accelerator model. And then finally, capital efficiency. So the options available for CDMOs and cell line development companies is really tremendous right now. The marketing around each of those customers is really quite, or each of these companies is really quite incredible. There's a lot of really good science out there, but that means that there is also a really high risk of making the wrong choice, or the consequences of making the wrong choice of who to partner with are quite high. We don't want people, or we don't want companies or drug developers to be partnering with inexperience, with cultural misfits, or even with the wrong cell line technology for their molecule in particular. The MAB accelerator eliminates this risk and allows a low commitment option to try before you buy with Syngene. And Suneet will now walk you through the technical strategies to reduce time to the clinic.
Thanks, Sam. Thanks for in that nice introduction. So let me tell what is SynWeave. SynWeave is Syngene's next generation cell line development platform. Basically, it's created fundamentally to change how early biologic development is done. The platform is designed to shorten development timeline, increase productivity, and most importantly, improve manufacturability, thereby reducing scale-up and downstream production risk. At the core of SynWeave is the integration of transposon-based gene integration technology with Syngene's deep expertise in upstream process development. This combination allows us to generate highly productive and stable clones more efficiently, and also while simultaneously evaluating those attributes which matter for scale-up and manufacturing. Our intent with SynWeave is very clear. It is not just first cell line development, but it's about first CLD along with early data-driven manufacturability signals, so that we Syngene help our partners in selecting the right clone early in order to avoid any late-stage surprises and move into scale-up with confidence. We are talking about our platform is faster, but mechanistically, how does it save time? So in order to understand, we have to understand what is called position effect. In conventional cell line development, where a gene is integrating into a genome, that strongly influences how well it is expressed. And if integration occurs into a transcriptionally inactive regions, we all know expression can be very low, unstable, and even silenced over time, leading to high clone-to-clone variability. How do we address that? So CNWIV addresses this through transposon-based semi-targeted integration, which efficiently inserts the complete expression cassette into a transcriptionally permissive regions of the genome. This significantly increases the proportion of productive stable clones and reduces the need for large-scale screening, which directly saves sometimes from our CLD. So what does it translate into saving of the timeline? This slide summarizes how CNWIV compresses the overall cell line development timeline to just 9 weeks from day of transfection. We start with gene synthesis and vector transfection, which is comparable to any standard platform. Where we really differentiate, it is the pool generation and clone generation phases. Pool generation and assessment is completed within only 3 weeks' time, and clone generation and further assessment are also accelerated. Overall, this results is about 7 weeks reduction in the CLD timeline. But importantly, this speed does not come at the expense of robustness of our platform. CNWIV has been successfully applied across multiple molecular formats: monoclonal antibodies, fusion protein, pi-specific, complex protein, and also enzymes. So the net impact is faster progression into downstream development and earlier value realization for our partners. So what happens within that 9 weeks of timeline? Basically, we do optimize our gene of interest, and then transfection happens. Based on the selection from the stable pool, we can go into monoclonality. We all understand the monoclonality assessment is a regulatory requirement, and we use cytin and solentium, which captures the image of monoclone, providing with the confidence the single cell clone which is coming is indeed a monoclone. We use octet system to measure the titer at high throughput level for IgG or FC fusion kind of protein. When you have selected the top clones based on the titer, it goes into AMBER system in order to identify the best top 5 clones. We are not only looking into the titer, we are also looking into the quality attributes which we can differentiate based on size, charge, glycan, and also activity to select the top 5 clones. Those top 5 selected monoclone goes into stability studies up to 60 to 80 generations, and we also carry out a copy number analysis by ddPCR. So this practical takeaway from this slide that as we data-driven clone selection reduces the risk of advancing any fast or fragile clone. So now I think all of us have seen that speed is great, but let's talk about titer and scalability. This slide illustrates how CNWIV delivers the stepwise improvement in productivity as program progresses throughout the development. We start at the cell line development stage, where baseline titer between 4 to 7 grams per liter has been achieved. And from there, initial upstream process development provides an additional 1.5 to 2-fold increase in the titer, generally reaching between 7 to 9 grams. We also have an intensification, N-1 intensification available if we have to increase the titer further, which we see a further gain taking titer up to 9 to 12 grams per liter. What's important to understand is that these improvements are not isolated optimizations. They're integrated enhancement built into our platform. So for clients, how does it matter? This translates into faster timelines, lower cost of goods, and greater confidence that productivity gain will carry forward into scale-up and manufacturing. Now let us talk about some metabolic behavior which is a often hidden risk at scale up. So you all understand lactate and ammonia are the crucial indicator of cell health and metabolite states. The fact that we see a conserved trends across clones tells us that the underlying cellular metabolism is behaving consistently without any unexpected divergence. This consistency is very important because it suggests that the clones are not developing any hidden liabilities that could emerge later part of the development. When clones behave similarly at the metabolic level, we all understand that there'll be enough greater confidence that scale up and progression into later stages will be more predictable and inherently lower risk. Another bottleneck, what we all understand, is the selection stress. How do we handle that? This slide again illustrates how cell pools recover following the antibiotic selection process across different molecule types. And you can see also the selection conditions. What's important here is that we consistently observe complete recovery within about 7 to 8 days. That rapid recovery is a strong indicator of cellular resilience and directly contributes to shorter overall development timelines, allowing programs to progress more quickly throughout early development. Now let's see some data what we have obtained for IgG kind of molecules. This is a case study for IgG1, and what you can appreciate that we observed our CNOE platform that 70% of the clones what we screen having high titer. So that during this platform, actually, lot of clones need not to be screened to get a better producer. What does it signify? It saves a lot of time and the cost of the raw materials, which can actually make your cell line development cost very reasonable. On the other hand, you also would appreciate that using our CNOE platform, we have obtained between 3 to 5 grams per liter titer for IgG1 kind of molecules without any optimization, as shown on the right side, and QP was also very high. Similar data we have obtained for IgG4 too. Again, 70% of there is a greater population shift towards the high producer. We have seen 70% of them are having a good titer, and the titer we have obtained for IgG kind of molecule is also 3 to 6 grams per liter titer. So you can clearly see that CNOE platform, there is no need to screen multiple clones to get a better producer. So you all understand that we are getting a good titer from the amber, but strong clones must also scale. So let's address the manufacturability across scales in this slide. So this slide highlights the consistency of the process as it moves across platform, from sec flush to amber 250 to 10 liter glass bioreactor, even up to 50 liter scale single-use bioreactors. What's striking is that how closely the growth and viability trajectories overlap across scales, indicating that the cells respond in very similar way regardless of the scale. On the right-hand side, you can see that the titer accumulation pattern are also very well aligned across different scales, ultimately reaching to the comparable final titers. This clearly tells us that the productivity is being preserved as we scale, rather than reducing at the larger volumes. So equally important for us to understand that replicate runs even shows minimal variability, which confirms strong batch-to-batch reproducibility. Taken together, CNOE platform across scale demonstrates a highly reliable and scalable process, giving us confidence that what we see at the small scale can be translated predictability into a larger manufacturing environments. So we talk about speed, we talk about titer, we talk about scalability. What about quality when we move from pool to clone? The key message on this slide is that as productivity increases from pool to clone across different molecules, what you can see on the left-hand side, the product quality remains very consistent. While we see expected increase in the titer during this transition, the quality attributes do not drift. On the right-hand side, we are showing both the data for the charge profile and monomer content. And you will notice that this quality parameter remains well aligned between clone and the pool stage. This consistency is very important because it tells us that higher productivity is not coming at the expense of product quality. Because of this predictability, it is quite possible to initiate toxicological studies directly using pool-derived material. This capability can significantly expedite the development timeline by removing the need to wait for the final clone selection before starting your tox work. Why this matter is so important? Because predictability quality behavior reduces your late-stage surprises, particularly as the programs moves into tox material generations and IND-enabling activities. Ultimately, it is lowering your developmental risk and accelerating any decision-making. So we all talk about the quality, titer, timeline, but there is another most important part. How about stability? Because stability is another major de-risking lever across generations. So this slide highlights the stability of SynWave clones across generations. On the left-hand side, you can see that high proportion of the clones maintain stable productivity even at the later generations, indicating strong genetic and phenotypic stability. On the right-hand side, across different molecule types, we observe consistently high clone stability from early to late generations. Importantly, to understand that this stability is accompanied also by homogeneous product quality, which will be discussed in the next slide. That tells us this platform is not just producing isolated high performance, but a robust and also consistent clone population. Now let's talk about the quality. As I told, this is the product quality holds across generations. Across generations, this slide is showing high degree of consistency in key quality attributes, including mainly SEC, charge variant, glycan profile, and also titer. What this tells us is that both productivity and product quality remain stable as cell line progresses over time. The key message is that from stability across generations directly support long-term manufacturability planning. When productivity and quality remain constant over time, the risk during scale-up and tech transfer and extended manufacturing campaign is significantly reduced, giving teams greater confidence that programs advance toward late-stage and commercial manufacturing. I think I have one more case study why we want to showcase that SynWave is a much better to any other standard platform. From a CDMO and client ROI perspective, this comparison clearly highlights the value of SynWave brings to the developmental programs. With SynWave, we are able to screen a significantly reduced number of clones, what I have already discussed earlier, while still identifying top performing candidates. That immediately lowers the developmental effort. Fewer clones to characterize, fewer experiments to run, and less time and material consumed, directly reducing the cost of goods and also overall developmental spend. If you look at the headline numbers, the best SynWave top clone reaches a titer of around 5.7 gram per liter, compared to about 2.5 gram per liter with a standard platform. Achieving more than a twofold improvement without expanding the screening funnel is a really major efficiency gain. For clients, this translates into a faster timelines, lower CMC risk, and early value realization. For Syngenes, what does it matter? It means delivering high quality, high productivity cell lines with fewer iterations, enabling programs to move more rapidly into process development and scale up and manufacturing. So let me summarize SynWeave, the value proposition in one slide. To summarize this, SynWeave is about high titer clones, high manufacturability, and shortened timelines. The next generation platform for speed, titer, and quality. So Sam, over to you to talk about the operation strategies now.
Thanks, Saneet. So in the final slide, I'll just take you through these operational strategies or ideas that we can use with a CDMO partnership for moving quicker to the clinic. I mentioned earlier that the industry typical timeline from gene sequence to clinical GMP supply is around 18, maybe 24 months. And there are multiple variations of this within the market, but Syngene's base timeline for an antibody is 9 months to drug substance supply, and then 10 to drug product. To reduce this even further, we have a number of operational strategies that we can employ or take advantage of, depending on specific customer needs, the molecule in question, or the regulatory strategy that's being employed with that particular molecule. So when shopping for a CDMO or when looking for a contractor to partner with, it might be worthwhile just thinking about some of these as options for that program, for your program as well. Removing an engineering batch, which is a practice run ahead of a GMP batch or GMP manufacture, can save around 10 to 14 weeks of labour time, but it will only be the right decision if the manufacturer has a really strong right first time manufacturing record. Now, Syngene's is 100% right first time manufacturing batches. This record is only achievable through rigorous scale-up strategies with reliable and reproducible small-scale, pilot or mid-scale scales of the final manufacturing production scale. Failing a GMP batch might result in an additional 6 to 8 weeks added to that timeline, but only in favourable situations will it be 6 to 8 weeks. If the manufacturing facility is booked fully, you might be looking at months of waiting time for the next available slot, plus those 6 to 8 weeks. So really understanding how you can minimise the risk of moving into that first GMP batch for clinical supply is really important. Speaking of which, when additional work is required, simplified business practices are really critical. When change orders are needed to a contract, how smoothly can they be executed? How quickly can that work begin? Is the development team that you're working with already going to be redeployed while you negotiate contracts? And then finally, a very popular approach right now is to generate material for toxicology studies or animal studies as early as possible in that timeline. Syngene can perform toxicology material supply runs during cell line development. So during that stage that Saneet talked about where stable pools are generated, we can take those pools and generate larger materials of it, of the material as a drug substance for animal studies. And that concludes the presentation. Thank you for listening.
As a reminder to our audience, you can go ahead and type in your questions for our speakers now. So our first question is, I'm developing a bispecific antibody. Does the MAB accelerator apply in this case?
So I can take that one. It more than likely does, yes. MAB and everything else. Accelerator did not quite roll off the tongue. So we went with MAB accelerator, but we are open to discussing all formats of proteins under this.
All right, and our next question is, what's the genetic stability data beyond 60 cell generations, and have you tested to 80 or 100 CGN?
Yeah, I can take it up. We have gone up to 80 generations and beyond for some of the client, and 80 generation stability is also quite good. We don't see any drop. We are yet to get any request to do the generation stability up to 100. But yeah, we have data till 80 generations.
And what's the clonal diversity after single cell cloning, and how many clones do you typically screen to get your top 5?
It's a very interesting question. So if you see what I have explained, that we are doing a bulk pool. So clones coming from the same bulk pool are usually less diverse. We do not need to screen. So, and because of the transposon-based technology, this is quite consistent. So we generally screen 500 clones to arrive to top 5 candidates.
And how do you define risk sharing contractually, and what happens if the cell line development fails to deliver a clone meeting the target product profile?
That is a good question. So our scientists and your scientists would have a real deep conversation up front to agree on what success is possible and what's likely to be the outcome. If we don't hit those targets, then there is nothing to pay from your side. However, Syngene will very likely try and do better, right? So we'll probably try and offer to do better next time, or try something different, or work with you to get to the results that you're looking for. Nothing to pay.
Great. And our next question is, what host cell lines do you offer, and what is your selection system?
Okay, I'll take it up, Sam. So basically, this is a CHO K1S cell line. This is not a GS knockout cell line. This is CHO K1, and we use antibiotic-based selection for our cell line development.
And maybe related to that question, what method of cloning is used in SynWeave?
So basically, we are using Cytina, image-based Cytina and Solentium for monoclonality. Using Cytina, we drop one cell into the well, and we take pictures from the day 0 to day 1, day 2 using Solentium. So that confirms the monoclonality. And we provide a monoclonality report as part of the regulatory submission.
All right. Well, it does look like we are running a bit short on time. So if we don't get to your questions, we'll be passing them along to Sam and Sunit, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, do you offer biosimilar development, and what are timelines?
I can take up the, so we do develop biosimilars. We have already developed a couple of biosimilars using our platform. And timelines we can discuss separately about developing, because you can imagine this is, you have to meet the quality parameter. We can talk more about the timeline. Do get in touch. We'll talk about it.
Get in touch.
All right. Well, Sam and Sunit, thank you so much for joining us today. We appreciate your time.
Thank you. Thank you so much. I did want to end with just one invitation to the audience, and that is that we will have a fairly large team at Bio in San Diego in June. If you are attending, or if you're in the area, please come by and say hello. I just need to check we're at booth 4907. Please come and see us.
All right. Thank you so much. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye-bye.